Okay, let's finish out our notes. I can't exactly remember where we left off, so I'm just going to start at the end of this second page of notes. We're at the bottom of the page that says Mendel, and we're going to finish that. If you've already done this, then just bear with me, and I'll get to the next page. So we started with two purple flowered pea plants that are true breeding, which means that they have two alleles that are the same. And in this case, the two alleles are dominant. We don't quite know that yet. Mendel hasn't quite discovered that, but that's what he's figuring out with this process. So plants in a true breeding strain, when crossed together, produce offspring that are just like their parents. Okay, so that's why we have two purple flowered plants that when crossed, they end up with four purple flowered plants. So all four of those are purple flowered. Okay, so an example also would be a cross between two true breeding white flowered plants, which would then produce all white flowered plants. Okay, moving on to the next page. If I can find it. Okay, so what then happens? What if you cross two different breeding strains, which basically means now I'm going to take that true breeding white flower plant and the true breeding purple flowered plant, and I'm going to cross those. Because we know if we do purple and purple, we get all purple offspring. If we do white and white, we get all white offspring. But what happens if we take a purple and a white? What happens? So Mendel took two true breeding strains, white flowered pea plants and purple flowered pea plants, and he crossed them together, and all of the offspring ended up being purple flowered. Why is that? Well, this helps. We realize in this point, or Mendel realized, okay, there's got to be some kind of dominant trait going on. So, and important to note is that these flowers weren't like a light purple or a pink color. They were the same exact purple as the parent. So they were not light purple or less purple than the parent plant. So let's identify our phenotypes and genotypes. So our phenotype for this white flowered pea plant is the white flowered color. The phenotype for the purple flowered is purple flowered. Why? Because that is the physical characteristic. Remember, phenotype, which you can't see what I'm writing there. Phenotype is the physical characteristic, right? Genotype is what we write with the letters. So our genotype for the white flowered plant, white here is not dominant, so we're going to use our lowercase f's. And the genotype over here, this is the dominant trait, so we're going to use uppercase f's. And when it's two of the same kind of uppercase or lowercase, it's called a homozygote. Homozygote. It's the arrow on both sides. Okay? So we have a homozygote dominant plant here and a homozygote recessive plant here. We're crossing them together. That essentially looks like, if you were to write it side by side, it'd be FF crossed with little f. So big F crossed with little f. And we're going to learn how to do a Punnett square on Wednesday, and a Punnett square will help us determine the ratios. And I'll probably show you at the end of this video what that looks like. But what we do know is that when he took this white flowered plant and this purple flowered plant, he ended up with all purple flowered plants. And none of them were light purple or pink. They were all the same color as the parent purple plant. Okay, so let's write what the progeny. Progeny means offspring, if you need to remind yourself what that means. Offspring. So the progeny of the purple plants. The phenotype is purple flowered. All the progeny are purple flowered. And the genotype, all of these plants are going to have the genotype big F, little f. Why? Let me show you. So we're crossing big F with little f. Okay? We do what's called a Punnett square. And I'm going to show you how to do this. We're going to do it again on Wednesday, but I'm going to show you ahead of time so that you can have an idea. So we're crossing this, and we're going to take one allele from the mom and one allele from the dad, and there's many combinations. You could do a big F with a little f. You could do a big F with a little f. You could do a big F with a little f, or you could do a big F with a little f. It's just showing, it's almost like factoring when you're doing math, where you're watching each one multiply against the other. So this results in all of these offspring to be what is called heterozygous. The hetero means different, and that's in reference to the different capital and lowercase. Okay? So that's our genotype, is big F, little f. Now, this is called the law of dominance, because Mendel found that some alleles are dominant while others are recessive. An organism with one dominant allele will display the effect of that dominant allele. So that's what Mendel figured out, but he didn't know some other things that we determined later. So scientists today use the term homozygote for an organism with two of the same allele. True breeding organisms are always homozygous. And heterozygous or heterozygote is the term used for an organism with two different alleles. This is called the law of dominance, when if there is a dominant allele, the capital letter, that one is always going to be the trait that shows up if that is the allele of that organism. The only way it is the recessive trait is if there's two of the recessive alleles. Okay, that's that page. Let's flip over on the back and do this last page. This is the last page we're going to do for notes, and then we'll do the rest on Wednesday. So what happens if you cross two heterozygotes? So now let's say we take two of these and we cross them. Remember, those are the heterozygous ones that are big F, little f. So what happens when we cross those? So let's put some words to that. Mendel took the progeny. Remember, progeny means offspring. Mendel took two purple plant progeny from his first cross, and we called these the F1 plants. This is the F1 generation. We started out P1. Now we're at F1. P1 is the parents' first generation. Now we're at basically the progeny's first generation. So that's F1. And we cross them. The progeny, called the F2 generation, that's when we take two F1s and cross them, had 3/4 or 3/4 purple flowered and 1/4 white flowered plants. Mendel concluded that while Mendel concluded that the white allele was carried by the F1 plants and could still be passed to the next generation. And each plant must be receiving one allele from each parent in the parent's gametes. Remember, the gametes are the cells that were created during meiosis. They are the sex cells, the sperm and the egg that come together. That's why you get one set from mom and one set from dad. Okay, the phenotype. So the phenotype of these F1 generation plants that we're going to mix, remember, we're going to take two of those purple flowered plants from the last cross, and we're going to cross those. So the phenotypes are both purple flowered, right? Because phenotype is the physical characteristic. Okay, purple flower, purple flower. Now, the genotype is big F, little f for both of them, big F, little f. Now, if you were here, I would ask you to tell me, is it homo or heterozygous? So this is going to be heterozygote. Zygous is the adjective. Zygote is that it's a noun. So we're calling this a heterozygote plant. So it's heterozygote because it has one allele of each kind. So it's the different, big F and a little f. If it was homozygous, it would be little f, or big F, big F. Okay? Now, notice down here what we have. We ended up with 1, 2, 3 purple and 1 white. Interesting. I can do a Punnett square for that in just a minute, but let's go ahead and write the words to this. So the phenotype that come out of this cross is we've got this phenotype for these three is purple flowered, but one of them, and we'll see this in a second, is big F, and 2 of them are big F, little f. And the phenotype for this white flowered plant is white flowered. Remember, phenotype is the physical characteristic. The genotype is the letters. What alleles do we have? And the only way that we can have white show up at all is if it's 2 little f's, little f, and this is homozygote. And this one right here, because it's big F, little f, is heterozygote, and this is homozygote. Now, if it's homozygote, if it's 2 big F's, it's homozygote dominant. If it's little f, it's homozygote recessive. All right, so what did Gregor Mendel learn? He figured out that organisms get one allele from each parent, and during gamete formation, the alleles for each gene segregate from each other so that each gamete carries only one allele for each gene. That's why when we looked at meiosis, In meiosis, we end up with the 4 daughter cells, and each daughter cell has one allele of each type because they split. And then when a gamete from the mom and a gamete from the dad come together, so the egg and the sperm come together and they form a zygote, those alleles come together and now they have one set of alleles from mom and one set of alleles from dad. And that's why dad contributes something to hair color and mom contributes something to hair color. Dad contributes something to eye color, mom contributes something to eye color. And that is how you get your traits that are contributed. And then there are just certain traits that are dominant and certain traits that are recessive. And that's what determines what shows up in the offspring. So things that Gregor Mendel didn't know is that scientists today know how meiosis works, but he didn't at the time. And they know that each gamete gets one chromosome from each homologous pair. This explains why each gamete only gets one allele from each gene. All right, quickly, I want to do the Punnett square for this cross, this F2 generation cross. So we're gonna now take 2 heterozygotes, big F, little f, and cross it with big F, little f. Hold on, you can't see that. All right, right there. So this is the cross we're gonna do. This is big F, and these are little f's here, okay? So I'm gonna make my Punnett square. And now you can see we're going to carry this across, these F's across, and we're going to carry these F's down. So this one is going to have 2 big F's. This one is going to have a big F and a little f. This one is going to have a big F and a little f. And this is going to have 2 little f's because I'm carrying, right? Does everybody see how I dropped those down and across? So now we end up with 1 big F, 2 big F, little f's, and 1 little f, little f. The 1 big F equals a purple plant, right? Because it's got the dominance. This is called homozygous dominant, and it's purple flowered. These are heterozygous dominant, and it's purple flowered. And this is homozygous recessive, and it's white flowered. Okay, so we'll do more of that on Wednesday. That is the rest of your notes for today. Sorry we didn't get to finish it in class, but we were having good conversation about genetics. Make sure you should be able to do both pages of homework with these notes now that we finished them. And remember to either take the homework quiz or link them on Google Classroom, depending on what the instructions say to do. The dominant and recessive allele homework page is a homework quiz, and the parent clues one is you take a picture of it and drop it back into as a file back onto the link for the assignment. Okay, if you have any questions, please either email me or do a remind text to me, and I will try to answer it, or call a friend and kind of get some input. Please do not just share your answers with each other. I want you guys to get an accurate view of what you understand, what you don't understand. There's no way that I can help you as a teacher know what you understand if you are just calling a friend and asking them for their answers. So, see you on Wednesday.